linkage, crossing over are the most discussed topic in genetics. Today's session we will discuss a very basic idea of crossing over and genetics, and in the next video we will learn that what are the types of crossing over and gene mapping. So in that case, first you need to remember that crossing over is only occur in पैकाइटिन स्टेज ऑफ प्रोफेज 1. तो माइटोसिस में क्रॉसिंग ओवर नहीं होता है और मियोसिस में क्रॉसिंग ओवर अगर होता है तो वो बस प्रोफेज 1 में और कौन से स्टेज में, वो पैकाइटिन स्टेज में होता है. दैट यू शुड रिमेंबर. अभी हम लोग जो क्रॉसिंग ओवर का जो बेसिक आइडिया है वो लेके काम करेंगे. फर्स्ट ऑफ ऑल, हियर is a chromosome. 2 genes are present, that is, I can locate them. There is one chromosome and another chromosome is that, and genes are small a, small b, small a, small b. Now, one thing that you should remember, they are actually homologous chromosome. This is a homologous chromosome. क्यों? क्योंकि यहां पे जो 2 क्रोमोसोम्स हैं इसमें ये जो है वो सेम है, एक दूसरे का रेप्लिका है, और ये भी और एक होमोलोगस क्रोमोसोम है. That is also a homologous chromosome. Now, that specific chromosome and this chromosome is known as the sister chromatids or sister chromosome. So it's sister chromosome, this is also sister chromosomes. So this chromosome and this chromosome is called the sister chromosomes or sister chromatids, and this chromosome and this chromosome is known as the non-sister chromosome or non-sister chromatids. So this chromosome is known as sister chromosome and this chromosome is known as a sister chromosome, and this chromosomes are known as non-sister chromosomes. यहां पे याद रखना होगा कि जो क्रॉसिंग ओवर है, क्रॉसिंग ओवर बस नॉन-सिस्टर क्रोमाटिड में ही होगा, सिस्टर क्रोमाटिड में नहीं. ये सिर्फ नॉन सिस्टर क्रोमोसोम में ही होगा, नॉन सिस्टर क्रोमाटिड में ही होगा, सिस्टर क्रोमाटिड में या सिस्टर क्रोमोसोम में ये नहीं होगा. तो ये कैसे हो सकता है ये हम लोग देखते हैं. सी, सो अगर ये दोनों में क्रॉसिंग ओवर होता है, अगर ये दोनों में क्रॉसिंग ओवर होता है, सो अल्टीमेटली व्हाट वी विल गेट इज दैट इज द सिस्टर क्रोमाटिड, that is another sister chromatid, and now chromosome will, crossing over will occur, that is so that is one crossing over. Another crossing over is so that is A, that is B. Here it's small a, here that is small b. So here we can see that that is the point of crossing over. That is the point of crossing over जहां पे मेनली ये क्रॉसिंग ओवर हुआ है, और ये जो है वो एक रिकॉम्बिनेशन भी है. और यू कैन से दैट साइट ऑफ रिकॉम्बिनेशन. रिकॉम्बिनेशन मीन्स व्हाट? रिकॉम्बिनेशन मीन्स कि recombination, that means two different chromosomes combine. Two different chromosomes will combine and produce a new type of chromosome where two segments of chromosomes are present in a single chromosome. So that is crossing over, and after that what will happen? We will get the gametes like A, B, then the recombinant one, another one, that is also a recombinant, and the parental chromosome. Now in this case we can see that these are the gametes, that will be produced. So what are the gametes? See, this chromosome and this chromosome is the parental chromosomes. These are the parental chromosomes. These are the parental chromosomes because you see that chromosome and that chromosome is exactly same like the parents. So it's called the parental chromosome. And these chromosomes, this one and this one, recombination occur in that chromosomes, and ultimately the products will be recombinants. यहां पे क्रॉसिंग ओवर हुआ है. द प्रोसेस इज कॉल्ड क्रॉसिंग ओवर and the phenomenon is called recombination, and the products that will be produced after the recombination is known as recombinants. Now if I want to calculate the crossing over frequency, now the crossing over frequency is crossing over Now, what would be the crossing over frequency? So, always remember that number of recombinant number of recombinant divided by number of total progeny into 100. Say we have total number of recombinant. Say we have 16 progeny. 16. We have 16 progeny and only say 6 recombinant. So the crossing over frequency will be 6 by 16 into 100. Then we will get the crossing over frequency. Now let's talk about linkage and linked genes. So first of all, what I would like to say is, when two genes are present in a single chromosome, two or more genes are present in a single chromosome, these genes are known as linked genes, and that phenomenon is known as linkage. So we can see, say it's a chromosome, and in that chromosome we can see that here is one gene, that is gene A. Another one is gene B. So these gene A and gene B is known as linked gene, and that condition, the phenomenon, is known as linkage. Now what we should know, that what factors are responsible for the strength of linkage. So factors. So three factors regulate the strength of linkage. So first of all, strength of linkage is proportional to 1 by crossing over. So that is the first factor. Second factor is it's proportional to 1 by distance between genes, and that is the second factor. And the third factor is proportionate 1 by independent assortment. So here three factors for the strength of linkage, that is 1 by crossing over, 1 by distance between genes, and 1 by independent assortment. So in these three cases we should know that if crossing over decreases, that means the strength of linkage increases. If the distance between gene decreases, strength of linkage increases. Same independent assortment decreases, so the strength of linkage increases. So say if here is one chromosome and gene A and gene B are linked. Gene A and gene B are linked, and the in between space, the distance between two genes are very low, then no crossing over. No crossing over or no independent assortment. So that means the strength of linkage will be higher. If the distance is low, then distance between the genes is low, that means no crossing over, no independent assortment, and the three cases, if these three factors are low, that means the strength of linkage will be high. If one chromosome, say this is a chromosome, this is another chromosome, and here we can see that two genes are present, that is A and that is B. Here another two genes, that is A and B. If say they are not linked, so what will happen? After that we will get the gene like A and B, that is a recombinant, that means crossing over occur. Another chromosome, that is another gene is B. So you can see in these cases, if genes are linked, no recombination, no crossing over, no independent assortment. But if genes are not linked, then what will happen? This crossing over, this recombination will occur. अभी थोड़ा सा अप्लाई करके देखते हैं कि लिंक जीन में ये काम कैसे करता है. Like here is one parent which is having two types of chromosomes. They are actually heterozygotes. So A, B, and that is A, B, and this parent we will cross with another homozygous, so that is a cross. So these are parents, and as we know that after that they will produce the gametes. So what will be the gametes? First gamete, so we can write that gametes what will be the gametes? So first gamete is A, B. Second gamete that I will get is this one, and the third type of gamete will come from this part. So from the law of segregation we get two types of gametes from here, this one, and as we know that they are homologous, so we get one type of gamete, that is this one. Now these are the gametes, and after that when we will cross it, that means we will cross these two gametes with this gamete. We will cross these two gametes with this gamete, and that we will get this one, and the another one that we will get is this one. So these are the F1 progeny. This one, these progenies are the F1 progenies. So one thing that is special in this case is both the cases, these chromosomes are actually the parental chromosomes. These chromosomes are actually the parental chromosomes. So what we will get, that is we will get the parental So that means if genes are linked, then we will get parental chromosome. We will get parental chromosome, and in the full process, in this full crossing, we have not seen a single crossing over or a single recombination. So we will write that no crossing over or no recombination. So in this case, if genes are linked, we will not see any crossing over or any recombination.